Thank you, Liam, for that introduction and for getting us started. Hi, everyone. My name is Amanshi Slaney, the Senior Director of Compliance and Regulatory Affairs, as well as DPO here at BioIVT. Welcome to everyone joining us today from wherever you are. BioIVT appreciates your participation. A little bit about me. I joined BioIVT 10 years ago. Prior to joining BioIVT, I oversaw regulatory for approximately 30 clinical trials at Central Florida Pulmonary Group. It was an honor to be a part of some of the early studies that brought relief to rare diseases like cystic fibrosis. At BioIVT, I currently oversee the improvement of various processes, including data privacy, integrating and harmonizing the various acquisitions from a regulatory perspective, and supporting the growth of the team and BioIVT. The C&RA team is a part of the larger quality organization, which is led by Matt Shorley, the VP of Global Quality. Global Quality reports directly to our CEO, Richard Haig. Before we dive into the various processes that BioIVT follows to ensure that researchers have peace of mind in using human biological specimens, I will briefly comment on the incidents and subsequent regulations that have shaped how we source and use HBS, some or all of which you're all already aware of. One of the landmark incidents and cases of our time is the story of cells grown from a biopsy obtained from an African American woman, Henrietta Lacks, in 1951, later called HeLa cells. Years later, and to her family's dismay, the ethics behind obtaining the biopsy, growing the cells, and then conducting research without informed consent from Lacks came to the spotlight. Organizations who use these HeLa cells are still compensating the Lacks estate and family members. In modern day, Lacks, as a result of the facts surrounding her story, may have even been considered part of a vulnerable population by an IRB. Note that there was no regulation at this time that specifically covered the use or of research involving identifiable biospecimens or identifiable data. A similar incident that influenced current regulations and ethical practices involve the lack of transparency to parents of newborn children in the state of Michigan on what would happen to blood spot cards collected from their infants. This involved the topic of consent and privacy, as these cards were kept for 35 years and then transferred to the Michigan Neonatal Bank for 100 years. Cases involving this issue are still ongoing. Finally, is an incident around members of the Havasupai Nation who donated blood for genetic studies on type 2 diabetes. However, they later found out that their samples had also been used in several other genetic studies concerning schizophrenia, alcoholism, and population inbreeding. The research topics were taboo in the Havasupai culture. The ethics in question were around broad consent and dealing with potentially vulnerable populations. The case was filed in 2004 and concluded in 2010 with monetary compensation and return of genetics. All of these stories played a major role in influencing the current regulatory landscape for the collection, use, transfer, and storage of biospecimens for research. One key regulation is 45 CFR 46, or better known as the Common Rule, which is overseen by the Office of Human Research Protection, or OHRP, of the U.S. Department of Health and Human Services. While the Common Rule applies mainly to federal agencies or institutions who receive federal funding, commercial or for-profit entities are captured by the provision included within 45 CFR 46 for IRBs and the definition of human research participant. Until the end of 2016, this regulation did not clarify the definition of an HRP to include identifiable biospecimens and identifiable data. The revised Common Rule went into effect in 2018. HIPAA, which stands for Health Insurance Portability and Accountability Act, is a privacy and security regulation overseen by US DHHS. While compliance with HIPAA does not directly apply to entities not dealing with insurance, the obtaining, using, transferring, and storing of data from research participants in clinical settings is heavily influenced by HIPAA. Although not specific to biological specimens, other privacy regulations, such as the UK and EU General Data Protection Regulation, GDPR, California's Consumer Privacy Act, CCPA, California Privacy Rights Act, CPRA, the Confidentiality of Medical Information Act in California, C&RA, and Protecting Americans' Data from Foreign Adversaries Act, PADFA, have also impacted the use, processing, and sharing of biological specimens and associated personal and sensitive data in research. I will discuss data later in the presentation. So how have we evolved as an industry since these incidents and regulations have come to pass? Today, research using biological specimens is a cornerstone of biomedical research, particularly in fields like genomics and personalized medicine. Large-scale initiatives such as the All of Us research program aim to collect biospecimens from diverse populations to advance precision medicine. Modern research emphasizes the importance of informed consent, privacy, and the ethical use of specimens. Researchers must navigate these complex regulations to ensure compliance and protect participants' rights. Advances in technology, such as whole genome sequencing or artificial intelligence, have expanded the potential uses of biospecimens, enabling more detailed and comprehensive studies. International collaborations and biobanks are becoming increasingly common, facilitating large-scale studies and the sharing of specimens and data across borders. To simplify the regulatory landscape internally, BioIVT has categorized all our HBS to these three categories: prospective, retrospective, or remnant, and postmortem. Prospective HBS would typically be collected specifically for BioIVT on behalf of our customers. It would include IRB or IEC approval, informed consent, comprehensive data, and may include compensation as approved by the IRB or IEC. Retrospective HBS would be collected for a different purpose, such as standard of care or other research, and would typically be discarded. This HBS would typically come pooled and or with limited data, as defined by HIPAA and anonymized. IRBs, IECs would typically provide a waiver of consent or an opinion of exemption for this type of material. Postmortem HBS is governed by the Uniform Anatomical Gift Act, UAGA, comes with mainly next of kin consent and sometimes donor directive, does not have IRB IEC approval or compensation. Here at BioIVT, we stay current and informed about each detail that influences our industry and strive to support researchers with navigating these changes. In addition to navigating the complexities of ethics and regulations involving HBS, the C&RA team, along with our quality counterparts, also oversee and support the various registrations, licenses, and permits and certificates. You can find direct access to some of these on BioIVT's website. It is now my pleasure to introduce Tracy Lynn, my colleague, to continue presenting important elements of ensuring compliance with the ethical and regulatory requirements of using HBS in your research. Tracy, the floor is yours.
Thank you, Amanchi. I joined BioIVT back in 2017 when Asteran was acquired. Prior to the acquisition, I worked for Asteran for 3 years as the regulatory manager for disease state. I formerly came from the University of Florida, in which I was the QA auditor and regulatory manager for the Cancer Center. I have 30+ years experience as a certified cancer registrar, and I'm currently pursuing my bachelor's degree in business. My main focus at BioIVT is on U.S. and international disease state tissues, which include the acquisition of Tissue Solution and Fidelis. The informed consent is a vital document in any research biospecimen collection. At BioIVT, the protection of human subjects is its highest priority. In order to achieve this, our consents will contain the following elements: a statement that the study involves research and the purpose of that research, a description of the procedures to be followed for research collections, a description of any foreseeable risk or discomforts to the subject, a description of any benefits to the subject or others which may be expected from the research. We also disclose an alternative procedure or course of treatment that may be available to the subject. We also state within the consent that the subject's information is confidential. For research involving more than minimal risk, an explanation as to whether any compensation and/or medical treatment will be available if an injury occurs. There's also an explanation of whom to contact for any questions regarding the research, and there will be a statement of participation that it is voluntary and refusal to participate will involve no penalty or loss of benefit to the subject. In addition, we also like to include a statement of the types of genetic research, and we also include those genetic risks associated to it. A statement that the limited withdrawal of the subject's specimen and data may not be destroyed upon request due to the sample or data have already been placed with the researcher. The consent will contain commercial use language identifying that the research may result in new products, tests, discoveries, which may have commercial value. The subject will not receive financial benefit from these results. From the research results, the consent will inform the subject that their specimen and data may be presented in scientific publications without identifying the subject. It will also include third-party language so that the subject is aware that the sample and data may be released to other research parties nationally and/or internationally. Lastly, data privacy regulation is mentioned within the consent in order to abide by local, national, and international regulations. For third-party consents, all consents are reviewed that the element of consent are present within their consent forms. If not, we advise the collection site to revise their consent and submit for ethical approval. I now have the extreme pleasure of introducing Latia Bullock, better known as T within our office. She will present the process of managing our BioIVT protocols, consents, and sites. Take it away, T.
Hello, and thank you, Tracy, for the introduction. My name is Latia Bullock, and I'm a Senior Manager of Compliance and Regulatory at BioIVT. I've been with BioIVT for almost 8 years, and my background is in clinical research and medical device regulatory. My primary focus here at BioIVT is enhancing and improving global regulatory processes as well as supporting customer needs. So, what is the process of managing BioIVT sponsored protocols, consent forms, and sites? First, the protocols and consent forms are written based on industry and company needs. Once the protocols and consent forms are written, they are then submitted and ethically approved by institutional review boards. These documents are reviewed and approved on an annual basis. So just to keep in mind that there are no changes to a protocol and or consent form for that year, the documents will remain the same version. Next, our sites. We have well over 400 US and international collection sites collectively, and these sites are managed through internal systems and processes. Internally, our legal team and development team ensure the full execution of contracts and agreements. Study startup documentation is provided, such as medical license, CVs, training records, and CLIA lab certificates, just to name a few. We liaison with US-based IRBs as well as international ethics committees to ensure site approvals and proper onboarding. We maintain a partnership with our sites and ongoing support for them by training and continuous improvement initiatives. In addition, communication is really key and essential for success. So, at BioIVT, we source material ethically and responsibly, and some key principles are informed consent. So, our colleagues discussed this already. Obtaining informed consent from donors is vital, and really clearly explaining the purpose of the research, the procedures involved, potential risk and discomforts, and the use of the specimens. And of course, donors must voluntarily agree to participate without any coercion. Transparency. So researchers must be transparent about how the specimens will be used, stored, and shared. Very important. Privacy and confidentiality. Protecting the privacy and confidentiality of donors is essential. So, personal identifiers are removed or coded to prevent unauthorized access to that very sensitive information. Regulatory compliance. We've heard it at the beginning of this presentation. Adhering to local, national, and international regulations. And this includes compliance with laws such as, of course, HIPAA in the US and GDPR in the UK and EU. Ethical review. Research involving biological specimens, of course, should undergo ethical review by an institutional review board or an equivalent ethics committee. So, of course, this ensures that research meets ethical standards and, of course, really protecting the rights and welfare of that particular donor. And we at BioIVT, we do our due diligence by adhering to these principles, ensuring written agreements are in place, and conducting audits and monitoring activities. So, to discuss auditing and monitoring, I'm handing it over to our colleague, Rashana. Rashana?
Thank you, Latia, for the introduction. As Latia has mentioned, I am the global auditor. I have joined BioIVT in September 2022. Prior to BioIVT, I did spend my time at Northwell Health and Mount Sinai Hospital, supporting clinical trials, and had the pleasure to co-publish a report which has contributed to the advancement of pediatric research in New York. I do have a Bachelor of Science in Health Sciences from Hofstra University, as well as a Master of Public Health and Health Policy and Management from NYU. My current focus is on supplier qualification and compliance with the intention of harmonizing audit processes across BioIVT. As the global auditor, it is my responsibility to ensure all BioIVT regulatory affairs and quality compliance standards are met when it comes to onboarding, auditing, and continuously monitoring all human biological specimen suppliers. This is an essential function as auditing not only enforces quality reassurance to our customers, but it also allows BioIVT as a whole to continually improve our operations that will result in a positive outcome in terms of product quality. It is mandatory to perform audits, and there are numerous benefits, with the following being the most valuable and has shown to be the most effective in achieving high quality standards. First in being risk management. Risk management is essential within any organization, regardless of size or line of business. And mitigation is an ongoing process which requires monitoring and ongoing diligence to keep up with the changing landscape of industry standards. The first step of effective risk mitigation is assessing potential risks that were identified when conducting the audit. This process involves all designated subject matter experts to effectively develop a risk management plan or similarly a corrective and prevention action plan. This has allowed myself to achieve the results of developing a robust organizational improvement plan to successfully reduce the negative impact of the identified risk and ultimately comply to BioIVT regulatory requirements. As a highly regulated industry, it is crucial for human biological specimen suppliers to be in compliance with applicable United States as well as international legislations and regulations. As all of you may know, industry standards are evolving, and it is our responsibility to not only be educated on these changes, but to also enforce them. Best practices include obtaining the specific supplier's existing accreditations and certifications, performing on-site audits to validate processes in accordance with specific standardized laws and regulations, protect data integrity, and identify any non-compliance which may result in reporting to the appropriate regulatory agency in a timely manner, which my colleague will discuss later in this presentation. Nonetheless, risk mitigation and ensuring compliance will and have led to organizations, including BioIVT, adjusting their processes, whether it be minor or major, to promote operational efficiency to better reflect the organization's values and secure long-term success of producing high-quality products, which will not be achievable without transparent communication. In maintaining regular collaboration with the relevant department leadership teams, accountability is held, which promotes awareness of who among teams can mitigate risks, and allocation of additional resources, such as recruiting personnel or investing in technology, to resolve any identified operational inefficiencies. This allows the overall commitment to innovation and continuous improvement to be achieved. Now I will move on to yet another critical component for not only BioIVT, but the industry as a whole that aims to answer the question, what is the importance of an onboarding process for a prospective human biological specimen supplier? Risk mitigation should be initiated right from the start during the onboarding process. The process begins, of course, when a new human biological specimen supplier has been identified by either the sourcing team or another department that is involved with procurement. The ongoing diligence questionnaire will be distributed to the prospective supplier, which serves as a supplier verification check, in which it assesses the quality of products, the supplier's capability, and effectiveness of their manufacturing process. This questionnaire is reflective of quality management system standards and human biological specimen regulations, which include at minimum the FDA ISO 9001, HTA, IRB, CDC, GMP, GLP, and GCP standards. Once the onboarding diligence questionnaire and any additional approval documentation, which, as mentioned before, includes certifications, accreditations, essential standard operating procedures, the supplier QMS validation requirements, confirmation of their logistics process, and other high functioning operations critical to business continuity have been reviewed by both quality assurance and regulatory affairs and are deemed satisfactory, the supplier will move on to be 100% approved internally. Only then can legal agreements and IRB study submissions proceed to be established to define the expectations of both parties. Once the supplier is registered with the appropriate BioIVT departments and regulatory agencies, a site initiation visit is conducted by the regulatory affairs team. We ensure the newly approved supplier's clinical team, principal investigator, and applicable department team members are trained on the correct operational procedures and documentation requirements to collect human biological specimen according to the protocol study. Not only is the primary investigator aware of their responsibilities, but this visit also ensures all team members are aligned with the ethical principles and BioIVT practices as previously mentioned by Latia. Finally, a periodic on-site or remote audit will be performed to affirm the foregoing supplier's manufacturing processes, to maintain the existing partnership through frequent communication, and to confirm the supplier's conformance to not only BioIVT standards, but also to industry standards. To summarize, regulatory ethical compliance starts at the very beginning during the supplier onboarding process, which is then maintained through ongoing diligence and continuing monitoring efforts. This has resulted in long-term beneficial partnerships, which serves in improving synergies between regulators and manufacturing companies while incorporating risk-based approaches. It is now my great pleasure to introduce my colleague, Compliance and Regulatory Manager, Stephanie Shipley. I will hand it over to you, Stephanie.
Thank you, Rashana. Hello. I joined BioIVT in 2019 with their acquisition of Keystone and PPA Research. Prior to that, I worked as the clinical research coordinator at PPA and PMG Research, which is now Accelicare, where I coordinated many clinical trials in a variety of areas and sites. I have a bachelor's degree in applied human science and a master's degree in clinical nutrition from ETSU. At BioIVT, I focus on the donor center network in the US and the UK. I will be reviewing deviation and noncompliance reporting for BioIVT's sponsored research. As part of our commitment to protecting the rights and welfare of human subjects in research, BioIVT requires prompt reporting of major deviations and noncompliance from our site network. Noncompliance can be described as any variation and/or admission from the approved study protocol, IRB requirements, or federal, state, or local regulations that may affect the subject's rights, safety, or well-being, and/or the completeness, accuracy, and reliability of the study data. Noncompliance can range from minor deviations to major deviations. Minor deviations do not affect the participant's safety or the integrity of the research. Minor deviations often can't be avoided and include such things as documentation errors, missed study visit windows, or a study procedure conducted out of sequence. Minor deviations that do not meet the IRB Requirements for reporting should be documented and placed in the regulatory binder and reported to the IRB with the next annual report. On the other hand, major deviations are things that impact the safety of the participant, affects data integrity, increases risk, or decreases benefit to the participant. For instance, enrollment of a subject who did not meet the inclusion-exclusion criteria, breaching confidentiality in a way that could put participants at risk, failing to report an adverse event, or not submitting required documentation to the IRB. Deviations vary in their incidence and impact and should be reported accordingly. Investigators and their study staff are required to report major deviations and noncompliance to BioIVT and the IRB no later than 5 days after discovery of the deviation or noncompliance. The IRB promptly reportable information form must be completed and submitted by the site, and this report should include a complete description of the deviation or noncompliance, the date of occurrence and the discovery date reported to the IRB, if the deviation resulted in an adverse event or an unanticipated problem, and any corrective action. In addition to notifying the IRB, the investigator will be responsible for ensuring that corrective and preventative measures are in place and a signed and dated corrective and preventative action plan is provided to BioIVT. Depending on the details, major protocol deviations may be determined to be noncompliance. When noncompliance happens, it can be serious, intentional or unintentional, sporadic, or continuing. Serious noncompliance creates an increased risk to subjects, adversely affects the rights, welfare, or safety of subjects, or adversely affects data integrity. Continuing noncompliance is a pattern of noncompliance that could suggest the likelihood that noncompliance will continue and can be willful or even unintentional. Serious and continuing noncompliance can lead to repercussions of suspension or termination, loss of trust, reputational damage, or even legal action. BioIVT has noncompliance and deviation reporting requirements built into third-party contracts. But if you'd like more information about reporting requirements, feel free to contact me. Now I'd like to introduce my colleague, Tracy Sabra, who will be discussing harmonizing and integration.
Thank you for the introduction, Stephanie, and I'm happy to be here. Hi, everyone. I'm Tracy Sabra, and I joined BioIVT through the acquisition of Precision Med in 2023. I've been with Precision Med for 23 years, enhancing and developing various areas of the business, mainly from the operational side, as well as regulatory affairs. Since the acquisition with BioIVT, my primary focus is compliance and regulatory integrations of clinical protocols and harmonizing SOPs. Today, I'll be discussing integrations and the harmonizing process here at BioIVT. The Integrations Management Office at BioIVT sets the timeline for all integrations. The Compliance and Regulatory Affairs team then prioritizes these integrations based on the highest risk exposure. Once the timeline and priorities are established, integration and harmonization activities commence. If the new acquisition lacks relevant policies and SOPs, BioIVT's policies and SOPs are promptly established with the acquired company. The compliance and regulatory integration activities include harmonizing the sourcing network, IRB/EC protocols, consent forms, and relevant SOPs and policies. BioIVT focuses on integrations and harmonizing in order to better support our researchers. I will now pass this on to Amanshi.
Thank you, Tracy, for that great synopsis and bringing us nearly to a conclusion. For everyone still around, hang in there, we're almost done. The last topic I will touch on is data. Data integrity, data privacy, and data security. Maintaining data integrity involves implementing measures such as error checking, validation procedures, and strict security protocols to prevent unauthorized access or corruption. This is crucial for making informed decisions, ensuring compliance with regulations, and maintaining trust in the data. BioIVT has a dedicated data team responsible for managing and monitoring data. Data privacy includes policies and practices that protect individuals' data from unauthorized access and misuse. Data privacy regulations, such as the General Data Protection Regulation, or GDPR, in the UK and EU, mandate that organizations handle personal data responsibly and transparently. Driven by the rights of data subjects, this includes obtaining consent from individuals, providing them with control over their data, and ensuring that data is used only for its intended purposes. Data protection includes implementing physical, technical, and administrative controls to ensure the confidentiality, integrity, and availability of data. Key strategies include encryption, access controls, regular backups, and disaster recovery plans. Data protection is essential for preventing data breaches, ensuring business continuity, and complying with legal and regulatory requirements. BioIVT prioritizes data integrity, privacy, and protection, and are continuously working towards building a robust framework that safeguards not only our data assets, but with commitment to maintaining the trust of our stakeholders, such as our suppliers, donors, and customers. Thank you for joining us today. That concludes the presentation portion. On to you, Liam.